Every parent says their child is perfect, but do they really think that? What if science allowed them the opportunity to make the perfect child? Would you edit your child? With a new technology known as CRISPR-Cas9, the power to do this could become a reality. Approximately 120,000 to 160,000 babies are born each year with some sort of genetic birth defect. CRISPR technology could lower these numbers to minuscule amounts. But how? Let's head back to the lab to find out more about genes. As you know, every human has unique DNA. Every cell in a human normally has 23 pairs of chromosomes, made up of DNA molecules like this one. Short sections of DNA are called genes. The letters A and C are paired with their complementary bases T and G respectively. Genes contain information on what an individual will look like and also control many activities in our body with the help of proteins that are encoded by genes. These proteins are made by DNA making a copy of its code, A's, T's, etc., onto a type of RNA molecule, which is a molecular cousin of DNA and typically contains only one strand. So that's how our genes work. But how does gene editing work? CRISPR-Cas9, though in its early stages of development, could potentially offer the possibility of altering our genes, or even the genes of an embryo. CRISPR was developed after scientists researched how bacteria can build up a type of immunity to certain viral infections.
The way the system works is that if the bacterium is invaded by a virus, then part of the bacterium can incorporate part of the virus into the genome. And when it's re-exposed to that virus, then it produces a small RNA, and the RNA associates with the Cas9 protein, and then the RNA Cas9 protein binds to the invading virus and inactivates the virus. So essentially it's a mechanism through which bacteria protect themselves against virus. CRISPRs are used as a genetic tool to change the genome.
Scientists then realised that this mechanism could be used to treat an abundance of genetic diseases. To get a better idea into how this works, let's look at sickle cell anemia, a genetic disease which changes the shape of red blood cells, causing clogged capillaries, severe pain, and even organ failure. This disease is caused by a single mutation of one of the letters of code in the DNA. This sequence should read GAG, but instead reads GTG. So a programmed piece of guide RNA is inserted into the cell and guides the Cas9 enzyme to cut the DNA strand at a specific site. After the Cas9 cuts the DNA, a new strand carrying the correct code that was also inserted into the cell is used as a template to reform a healthy DNA.
You have the potential to use the technology and to abuse the technology, I guess. And that's the way it is with all technologies.
In theory, this technique enables the creation of a model person, although changing these genes may alter more characteristic about the person as a side effect.
Of course, you can change if you have a gene that affects eye color or hair color or whatever. If you can find genes or multiple genes that will affect intelligence, then CRISPR allows you to change and mutate those genes in the germline. But of course, that brings up the whole area of manipulating genes within the germline, because I think changing these genes can have consequences that we are totally unaware of.
If we eradicate mutations leading to genetic diseases, are we devaluing disabled lives? Will the wealthy be able to ensure their children meet the beauty standards of the day and maybe even enhance their child's intelligence, while putting the children of the poor at a further disadvantage?
Manipulating genomes for research is fine. That's obviously not terribly controversial. To use it for changing eye color or hair color or whatever, intelligence or so on, I think that's a much more complicated area. Negative impact, I think, like all technologies, if people begin to use it, trying to gain advantage for their children or their offspring or whatever, to affect the traits of all individuals and all the offspring of that individual.
Like any other new technology, a great deal more research and experimentation has to be done to fully understand how humans will react to it. But its possibilities for curing disease and even research are endless. But the debate goes on. If you could have been genetically edited, would you want to be?